friends, I am Ankita Tiwari, research scholar pursuing my PhD from Department of Pharmaceutical Sciences, Dr. H.S. Gaur University, Sagar. I am really privileged to discuss this very interesting topic of B.Pharm Vth semester entitled as Phase 1 Metabolism, which is scripted by Dr. Ram Kishore Agrawal, Dean, Department of Pharmaceutical Sciences, Dr. H.S. Gaur University, Sagar. Today, we are going to learn an interesting chapter. The chapter is divided into following modules. Module 1, Reduction of Carbonyl function. Module 2, Reductive reactions of Nitro group. Module 3, Reduction of Tertiary amine oxides. Module 4, Reductive dehalogenation. Module 5, Hydrolytic reactions. Introduction. Drugs may be metabolized by a wide variety of enzymes located throughout the body. Also, there is a wide variety of reactions that can be called metabolism. These reactions may be grouped into phase 1 and phase 2 type reactions. However, some have included phase 0 and phase 3 transport processes as part of the overall topic of metabolism. Commonly, there are 4 types of reactions involved in drug metabolism. These are oxidation, reduction, hydrolysis, and conjugation. Phase 1 metabolic processes include oxidation, reduction, and hydrolysis reactions, which typically provide functional groups capable of undergoing phase 2 reactions. The enzymes which catalyze phase 1 reactions are found in a number of subcellular components including cytoplasm, mitochondria, and endoplasmic reticulum. Although the liver is a major organ of metabolism, metabolic enzymes are found throughout the body. Reductive biotransformations of several compounds, example pentavalent arsenic and xenobiotics containing an aldehyde, ketone, disulfide, sulfoxide, quinone, N-oxide, alkene, azo or nitro group are often reduced in vivo, although it is sometimes difficult to ascertain whether the reaction proceeds enzymatically or non-enzymatically by interaction with reducing agents such as the reduced forms of glutathione, FAD, FMN, and NADP. Some of these functional groups can be either reduced or oxidized. For example, aldehydes can be reduced to an alcohol or oxidized to a carboxylic acid, whereas sulfoxides can be reduced to a sulfide or oxidized to a sulfone. In the case of halogenated hydrocarbons such as halothane, dehalogenation can proceed by an oxidative or reductive pathway, both of which are catalyzed by the same enzyme, namely cytochrome P450. In some cases, such as azoreduction, nitroreduction, and the reduction of some alkenes, example cinnamic acid, the reaction is largely catalyzed by intestinal microflora. The liver CYP dependent drug metabolism system is capable of reducing N-oxide, nitro, and azo bonds, whereas the cytosolic nitrobenzene reductase activity is mainly due to CYP reductase, which transforms nitrobenzene into its hydroxyl amino derivative. NADPH cytochrome C reductase is also able to catalyze the reduction of nitro compounds. These metabolic conversions may also be brought about by gastrointestinal anaerobic bacteria. Reductive processes that occur during the metabolism of xenobiotics involve either 1 electron reduction or a 2 electron transfer. Ionic reduction using a hydride occurs in vivo during the reduction catalyzed by NADH or NADPH enzymes, whereas one electron reduction releases a radical structure which may contribute to the toxic effect. Module 1. Reduction of carbonyl function. The understanding of metabolism is a crucial issue in the development and clinical use of drugs. Cytochrome P450 catalyzed oxidative reactions are the most common phase 1 reactions in drug metabolism. Carbonyl reduction is the major phase 1 reaction for an array of xenobiotics. Despite its importance, the reduction of the carbonyl group has been poorly studied. As a consequence, limited information is available on the carbonyl reducing pathways in drug metabolism. Carbonyl reduction refers to the reduction of an aldehyde or ketone moiety into a hydroxyl group. The reduction of a carbonyl group is a typical phase 1 reaction because it increases hydrophilicity and allows conjugation, that is phase 2 reaction, thereby facilitating the excretion of a drug. On the other hand, the conversion of a carbonyl to a hydroxyl group may have a profound effect on the biological activity of endogenous as well as exogenous compounds. Thus, carbonyl reduction plays a significant role in the activation or inactivation of drugs bearing a carbonyl group. Further, carbonyl reduction also represents a detoxification step because it decreases the chemical reactivity of aldehydes and ketones. Carbonyl reduction could in some cases lead to unfavorable bioactivation. For example, the carbonyl reduction of doxorubicin and daunorubicin represents the deactivation step of these anthracycline type anti-cancer drugs, but it is believed to be responsible at least in part for the anthracycline cardiotoxicity. Therefore, the prevention of carbonyl reduction may enhance the efficacy of drugs that are extensively metabolized via reduction, and it sometimes may even improve a drug's safety profile. Carbonyl reducing enzymes are NADPH dependent reductases that prefer NADPH over NADH. These reductases belong mainly to the aldo-keto reductases and the short chain dehydrogenases or reductase superfamilies. The HDR superfamily is one of the largest protein superfamilies, and the number of HDR members is constantly increasing. In humans, 75 genes belonging to the HDR superfamily have been found to date. The HDR enzymes manifest various catalytic activities such as retinol dehydrogenase, hydroxysteroid dehydrogenase, carbonyl reductase, or 15-hydroxyprostaglandin dehydrogenase activity. The majority of HDRs are localized to the cytosol, but some members are found in the membranes of endoplasmic reticulum, that is in microsomes, or in the membranes of the mitochondria or peroxisomes. The AKR superfamily comprises 15 human proteins belonging to the AKR1, AKR6 and AKR7 families. The largest AKR family in humans is the AKR1 family, which contains the aldehyde or aldose reductases. AKR1A1, AKR1B1, AKR1B10 and AKR1B15. HSDs AKR1C1, AKR1C2, AKR1C3 and AKR1C4. Steroid 5B reductase AKR1D1 and the testis specific protein AKR1E2. All of the enzymes reported to participate in the metabolism of carbonyl compounds are listed in table 1. Overview of CREs involved in xenobiotic metabolism. Protein names, subcellular localization, enzyme superfamily. Alcohol dehydrogenase, aldehyde reductase, cytosol, AKR. Aldose reductase, cytosol, AKR. Aldoketo reductase, cytosol, AKR. Aldoketo reductase, cytosol, AKR. Aflatoxin B1 aldehyde reductase, cytosol, AKR. Carbonyl reductase, NADPH1, cytosol, SDR. Carbonyl reductase, NADPH3, cytosol, SDR. Corticosteroid 11 beta dehydrogenase, endoplasmic reticulum, SDR. Dehydrogenase or reductase, SDR family, member 2, nucleus, SDR. L-xylulose reductase, membrane, SDR. The CREs from the AKR and SDR superfamilies show broad and overlapping substrate specificity. Thus, one compound may be a substrate of multiple enzymes from different superfamilies. For example, daunorubicin is a substrate of AKR1A1, AKR1B10, AKR1C2 and CBR. Another common feature of these enzymes is that they catalyze the reduction of both endogenous substrates and xenobiotics. Another characteristic of CREs is their ability to catalyze stereospecific reduction. Drugs undergoing carbonyl reduction. In recent years, a number of drugs have been reported to undergo carbonyl reduction. However, the knowledge of the carbonyl reduction of these drugs varies widely. The carbonyl reduced drugs have been divided into 3 groups according to the extent to which the participating enzymes have been characterized. Table 2. Group 1 consists of drugs that are substrates of CREs that were identified either after purification or by the use of recombinant enzymes. Thus, the involvement of these enzymes is unequivocally defined. The metabolism of drugs in group 2 is not as clear as in the first group. In vitro inhibition studies were performed with these drugs. Thus, the CREs participating in their biotransformation are not fully characterized. Moreover, the reduction drugs in group 2 has not been investigated in all of the subcellular fractions that participate substantially in their reduction. Therefore, crucial information on their metabolism is missing. Group 3 includes drugs with poorly described reductive metabolism. The reduced metabolites of the drugs in group 3 have been detected after administration. Despite the substantial use of these drugs in pharmacotherapy, the data regarding the CREs involved in their reduction are unclear or lacking altogether. Table 2. Drugs undergoing carbonyl reduction. Group 1 glucocorticoids. Group 2 nabumeton. Group 3 pentoxyphylline. Group 1 dimefluron. Group 2 warfarin. Group 3 neftazone. Group 1 tibolone. Group 2 epirazone. Group 3 oxycarbazepine. Group 1 norethindrel. Group 2 tolperazone. Group 3 fenofibrate. Group 1 votmanin. Group 2 bupropion. Group 3 ophthalmic beta blocker drugs. Group 1 benfluron. Group 2 metirapon. Group 3 fenofibric acid. Tibolone is metabolized by HSD by carbonyl reduction metabolism. Similar to tibolone, bupropion is also metabolized into its hydroxyl derivative. Similar to tibolone and bupropion, fenofibrate is also metabolized into benzyl hydrol fenofibric acid in the intestine via the single pass through the liver using enzymatic degradation. Module 2. Reductive reactions of nitro group. The nitro group is composed of a nitrogen atom bound to 2 oxygen atoms and a carbon atom. nitro compounds are very different from amines in their physiochemical properties because the nitrogen atom of the nitro group is not basic or nucleophilic. However, because of the electronic nature of the atoms, its composite atoms, the nitro group is relatively polar group. The most significant reaction of nitro groups both chemically and metabolically is reduction. The reduction of organic nitro groups is a well-known metabolic reaction, but the nature of the enzymes effecting this reaction is unclear. Westphal reported that the liver, kidney and heart are most active in the reduction of trinitrotoluene and that a succinic dehydrogenase preparation from beef heart is capable of affecting this reaction. However, Beuding and Jolliffe found that succinic dehydrogenase could not catalyze this reaction directly. They postulated that such substrates as succinate, lactate and malate regenerated diphosphopiridine nucleotide hydrogenase, that is DPNH, which in turn reduced trinitrotoluene catalytically through a flavoprotein. Purified xanthine oxidase can reduce organic nitro groups as well as inorganic nitrate. Liver xanthine oxidase reduced nitrophenols with DPNH as electron donor. Typically, nitro groups are readily reduced to the corresponding amines as illustrated in the example below. The reduction of the nitro group proceeds either by 1 electron or 2 electron mechanisms, that is given in figure 1. In addition, iron or other metals and reduced sulfur compounds can serve reductants for the non-enzymatic reduction of nitroaromatic compounds. Both the nitro group and the amino group are relatively stable. The sequence of reactions involved in reduction of the nitro group to the amine produces highly reactive intermediates. However, the nitroso and hydroxyl amino groups are electrophiles that can interact with biomolecules to cause toxic, carcinogenic and mutagenic effects. The 1 electron reduction of the nitro group produces a nitro radical anion which can be oxidized to the starting material by molecular oxygen with the concomitant production of superoxide. This futile cycle leads to the designation of enzymes that catalyze 1 electron reduction of the nitro group as oxygen sensitive. Enzymes from a variety of sources including anaerobic bacteria such as Clostridium species, facultative bacteria such as E. coli and Enterobacter species, as well as plants and animals catalyze 1 electron reduction of the nitro group. Reduction of the aromatic nitro group by the sequential addition of pairs of electrons is oxygen insensitive because no radicals are produced. Nitroreductases of this type convert nitro groups to either hydroxyl amines or amines by the addition of electron pairs donated by reduced pyridine nucleotides. The reaction pathway includes the nitroso derivatives, but they are difficult to detect because they are so reactive and unstable. The high reactivity of the nitroso and hydroxyl amino intermediates is responsible for much of the toxicity and carcinogenicity attributed to nitroaromatic compounds. Both structures can react readily with a variety of biological materials and they can also undergo condensation reactions. For example, partial reduction of the nitro group in the presence of oxygen leads to the non-enzymatic production of azoxy compounds as condensation products. The ease of reduction of the aromatic nitro group depends on the nature of the other substituents on the ring and on the reducing potential of the environment. Electron withdrawing groups activate the molecules for reduction of the nitro group, whereas electron donating groups make the ring more susceptible to electrophilic attack. In the ease of the nitrotoluenes, the probability of reduction increases and the probability of electrophilic attack decreases as the number of nitro groups increases. Figure 1. Reduction of nitro groups by 1 electron or 2 electron mechanisms. Ni-40MOX is metabolized by 2 electron mechanism into saturated open chain nitrile. Module 3. Reduction of tertiary amine oxides. N-oxide reduction has been reported to be enzymatic, non-enzymatic or both, aerobic or anaerobic, heat labile or heat stable, dependent on or not connected with xanthine oxidase or dependent on various cofactors. Non-enzymatic reduction of N-oxide has also been reported. Trimethylamine N-oxide can be reduced by iron, cysteine or reduced glutathione. Similarly, chlorpromazine N-oxide was reduced by Fe2 plus heme iron, also had an effect on N-oxide reduction. The N-oxides of NN-dimethyl-aniline, NN-dimethylaminoazobenzene and imipramine are readily reduced in erythrocytes or in oxygenated solutions of hemoglobin. Tertiary amine N-oxides were demonstrated to be reduced back to tertiary amines in the presence of NADPH or NADH. Most of the reductase activity was located in the liver microsomal fraction. The microsomal NADPH dependent N-oxide reductase activity was inducible by phenobarbital pretreatment, oxygen sensitive and inhibited by carbon monoxide, N-octylamine, 2,4-dichloro-6-phenylphenoxyethylamine, that is DPEA, and various other P450 inhibitors, suggesting the involvement of cytochrome P450 in N-oxide reduction. In addition to the microsomal reduction of N-oxide, the cytosol also appears to be a site for reduction of certain tertiary aliphatic amine N-oxides. The enzymatic reduction seems to involve xanthine oxidase and or cytochrome P450 in the liver preparations. The N-oxide reduction was blocked by allopurinol, an inhibitor of xanthine oxidase, and N-octylamine, an inhibitor of cytochrome P450 N-oxide reductase. The concurrent occurrence of N-oxide reduction and N-oxidation makes the investigation of in vivo N-oxide reduction to its parent tertiary amine difficult. The reversible N-oxidation of these tertiary amines and their further oxidation or hydroxylation may be accounted for the absence of N-oxide reduction pathway in vivo. Although N-oxides may undergo in vivo reduction, a reversible oxidation or reduction may also occur, and thus N-oxides may have long half-lives. Module 4. Reductive dehalogenation. Dehalogenation is the method of removing halogen, that is fluorine, chlorine, bromine and iodine from xenobiotics. There are three major mechanisms for removing halogens from aliphatic xenobiotics. The first, known as reductive dehalogenation, involves replacement of a halogen with hydrogen. In the second mechanism, known as oxidative dehalogenation, a halogen and hydrogen on the same carbon atom are replaced with oxygen, and the third mechanism of dehalogenation involves the elimination of two halogens on adjacent carbon atoms to form a carbon-carbon double bond. Reductive dehalogenation is catalyzed by cytochrome P450. The ability of cytochrome P450 to catalyze both reductive and oxidative reactions. Module 5. Hydrolytic reactions. Addition of water with breakdown of molecule is termed as hydrolytic reaction. Usually, hydrolytic reactions occur in blood plasma, example esterases, and inside liver. Among substitution reactions, ester and amide hydrolysis are common and often operate during detoxification processes. Both specific enzymatic and chemical hydrolysis may occur. Acid-catalyzed reactions may occur in the stomach and the kidney, whereas base-catalyzed reactions may be assisted by the alkaline pH of the intestine. A variety of hydrolytic enzymes, namely carboxyl esterases, choline esterases, and organophosphatases that hydrolyze xenobiotics containing such functional groups as a carboxylic acid ester, example procane, amide, example procaneamide, thioester, example spironolactone, phosphoric acid ester, example paraxone, and acid anhydride, example diisopropyl fluorophosphate. The hydrolysis of carboxylic acid esters, amides, and thioesters is largely catalyzed by carboxyl esterases, which are located in various tissues and serum, and by two esterases in blood, true acetylcholine esterase, which is located on erythrocyte membranes and is a dimeric enzyme, and pseudocholine esterase, which is also known as butyrylcholine esterase and is located in serum. Procane, a local anesthetic agent, is metabolized to its corresponding carboxylic acid derivative. Similar to carboxylic acid ester derivative, that is procane, phosphoric acid ester, example paraxone, also metabolizes into their respective phosphoric acid derivative.